A very good morning to one and all. Our speaker has joined with us today. Welcome back to the Scientific Lecture Series, Ocean Research Knowledge Series 2026, organized by Center for Ocean Research and National Facility for Coastal and Marine Research, Satyabama Institute of Science and Technology. So this ORKS 2026 is a continuous 30+ hour lecture series delving on the research insights from Center for Ocean Research and the National Facility for Coastal and Marine Research. Today we have with us our speaker, Dr. M. Ravi, who is going to be giving an expert lecture on shrimp aquaculture and animal health. Welcome, sir. Over to you.
Thank you, ma'am. Please allow me to share the video, my presentation.
Just a minute, sir. Mr. Taufik or Mr. Ram, if you are there in the meeting, please give permission to share screen.
Yeah, got it. Soumya is visible?
Your screen is not visible, sir.
Okay.
Please press share entire screen and then move to your PPT window, sir. Yeah.
Now?
Now it is visible, sir. Can open your PPT window. Yes, sir. And slideshow mode, sir. Yeah.
Now okay?
Yes, sir. It's visible now. Proceed, sir, please.
Yeah, wait a minute.
Stop sharing again.
Okay.
Yeah.
Yeah. Actually, today we are going to see about the research challenges in aquatic animal disease affecting shrimp aquaculture in India. So, before going to the topic, I'd like to introduce myself. I am Dr. Ravi, Associate Professor Research in Center for Ocean Research. Uh, actually, shrimp farming has become one of the India's most export-oriented industry. India export more than half a billion tons of shrimp annually, generally billions of dollars in foreign exchange. However, disease outbreaks continue to be the biggest challenge affecting production profitability and sustainability. So today I'm going to focus on emerging shrimp disease, especially the EHP management, recent bacterial disease. Actually, most of them know about the bacterial infection is like only the Vibrio, but we have a lot of bacterial disease. So this is what today we are going to discuss. Uh, Actually, the ship disease have changed dramatically over the last 2 decades. Earlier, we were mainly worried about the WSSV, means white spot syndrome virus, yellow head virus, TSV, okay, IHHV. But today's scenario is much more complicated because the farmers are mostly affected with EPO. Stop on it, Anand.
Download பண்ணலையா சார்?
டவுன்லோட் பண்ணிட்டுமா? 1 minute, 1 minute. அது
Download பண்ணிட்டு இன்னைக்கு ஓபன் பண்ணிட்டு ஸ்கிரீன்ஷாட் பண்ணிட்டு ஓபன் பண்ணு. Download பண்ணிட்டு இங்க வச்சுக்கோங்க சார்.
sometimes ஒரு வீடியோ ஒர்க் ஆக மாட்டேது. ஸோ
Share from there. Apologies, there is a slight technical issue. Our speaker will be sharing his screen shortly. Thank you for your patience.
now it's okay, moving.
Yeah, your screen is visible, sir. Please enter right to mode, sir.
Now it's full screen now.
I think there is a lag in your internet, sir. So I'm still able to see only the PowerPoint slide window. You can please enter the slideshow mode at the bottom, sir.
Now
Yes, now it's visible. Please
now
proceed. Yeah,
it's moving, right?
yes, sir, it's moving now. Yeah, thank you, sir.
Okay, thank you. So nowadays the farmers are they facing mostly on EHP. EHP is a enterocytosome hepatopene and Vibrio associated disease, and we have a lot of things. So today, firstly, we have to see about what is EHP infection. So it is kind of a microscopic parasite, and it is unlike WSSV. It usually does not kill the directly. Instead of it infect the hepatopancreases. So the hepatopancreases perform functions similar to the liver and pancreases. It is responsible for digestion, nutrition absorption, enzyme secretion, energy storage. So when EHP damage the hepatopancreatic cell, feed digestion decrease. Shrimp continue to eating, but nutrition cannot be absorbed efficiently. Therefore, the growth become extremely slow. This explain why farmers observe, but a large size variation within the same period. Some shrimp reach market size, while other remain very small. This phenomena is one of the biggest indication of EHP infection. So, the EHP uh, spread by spore form. Okay, the spore survive under harsh environment conditions, and transmissions occurs through infected seed, contaminated pond soil or water, carries such as crabs, polychaete, and other insects and fecal matters. And actually, the hepatopancreatitis it infect the hepatopancreatic cell. Then it will inject through the polar flagellum, and it will inject the spores, and it multiply inside the uh, cell. The practical method for EHV spore inactivation in shrimp pond. So how to uh, inactivate? This is mainly it is not like a theoretical presentation. It is more practical oriented, especially for farmer oriented presentation. So the EHP mostly involved in four different aspects like seed, soil, water, and carriers like shrimp, insect, birds like that. So from this infection, EHP infection, we need to control the pH before starting our pond management. We need to use lime or sodium hydroxide to control the infection. So we need to apply it to the pond. So how farmer may apply this pond? It is no, not plain. Sorry. So for lime application, we need to apply a lime and make it first. We need to apply the water into the pond and make it wet and apply the lime powder. And we need to keep 2 days with a high pH because of the lime solution, lime powder. The pH will be very high. So in this condition, after application of a lime treatment, we need to wait at least 2 days. After that, we need to apply water, and we need, before going for stocking, we need to check the uh, uh, pH. Otherwise, we need to do the proper conditioning, water conditioning. So many of the farmers, they won't do the pH checking and anything. So they directly go for the stocking. This is the biggest problem in the aquaculture industry. So the problems is like soil pH increasing and the calcium also very high. The labor cost, because they are applying the lot of lime into the pond, means it took a lot of uh, labor cost. So next one is sodium hydroxide. Sodium hydroxide is the next application study. So we have to apply 2.8 acres. We don't have actually the proper scientific evidence, but a delivery problem is we can spray through manual spray, and we can use tractor spray, and tanker spray also is available. So the advantage is it is very low cost because we can easily spray the some equipment. So we don't need manpower. So with very less manpower, so we can easily apply this high sodium hydroxide, and the concentration is very high, and it will increase the pH and corrosion. The next one is actually we have video. I'm not able to play now. I don't know. So the next one is potassium peroxide permanganate treatment. So we can apply with 15 ppm and 12 kg per 12 kg per off feet of water level. The equipment is we no need to any equipments. The necessary we can apply it, 50 to 100 ppm of uh, potassium permanganate. And for potassium permanganate treatment, we have a lot of scientific evidence to control the EHP infection in the pond. So they already suggested the chlorine application, soil, and water. What do for following the bore water, and how they are applying the solution for potassium permanganate. So we need to uh, uh, fill the half feet of pumped water, and 12 kg per ppm acres. Same day where we need to apply the water. So the bleaching will be the 53% over. So normal bleaching 6%. So this will be we need to keep it in 48 hours. After that, fill the water, and we can stock the animal. For some people, they can use the creek water or uh, saline water from the outside. So they must use the 12 kg uh, uh, of potassium permanganate per acre and spray through. And we need to wait for 48 hours, fill the water, and we need to apply 20 ppm of chlorine for reducing the pH. And uh, next one is the formalin treatment. The 200 ppm of formalin treatment also one of the key treatment solution for EHP infection. So we can do the spore activation inactivation by 95.33%, not 100% it is. So it, and we need to apply 240 liters per acre of the feet of water. We just fill the of the feet of water in the pond, apply the 240 liters of formalin solution in 1 acre. So it is not a formalin is not a commercial product. So we don't know about the exact concentration of the formalin. So the farmers may misuse the formalin solution. They don't know the because they don't know the concentration of product. So The main difficulty is the smell because the formalin is a very carcinogenic chemical, so it will make a very strong odor and irritation also. But in India, the some farmers they using formalin solution for treat the EHP. And resting pond means the if we do the shrimp farming, so we need to give the after harvesting the shrimp, we need to give some rest for the uh, pond because this rest will be the useful to treat the pond. If any infection is before that, so we must treat the uh, pond. After that only we need to go for next level of farming. So we need to wait at least 10 days after dechlorination. Conditioning the period is 10 days only, the microbial stabilization also. So after treatment of chemical treatment, we need to fill the water and we need to wait for some microbial uh, formation. And we need to balance the chlorine chlorine dioxide. So we don't have any data about this, but this is most important concern. We need to maintain this. And another thing is shrimp seed selection. So before buying any shrimp seed from hatchery, we need to confirm with the PCR and qPCR. And the same brood stock also we need to check with the fecal matter. With we need to collect the fecal matter and confirm with the PCR for EHP positive or negative. And next one is the common mistakes. What they do in farmers, they don't allow the exact water level, and if any low level of EHP is there, so it is difficult to identify. So the farmers may mistakenly buy the seed and they lose the culture. And the sodium hydroxide dose also is very biggest issue. So that's like mostly all the chemicals it is available in market, but we don't know the exact dosage. Dosage means the concentration of the chemical. So that also we need to take in this. So next level is next one is if any infection is come to the shrimp in this shrimp farm, first farmers may think it is WSSV or anything. So they want to check the WSSV by PCR or they can check the symptoms. If not negative, they must go for Vibrio infection. So it is Vibrio positive or negative or green colony or yellow colony. So what is the colony count? This is what the farmers may think about that. But we have a lot of bacterial infection in shrimp industries. So Vibrio-like bacteria, we have a lot of bacterial things. So especially for Photobacterium, yearly may form a diagnosed Vibrio infection. Today, many of these are actually caused by Photobacterium. The clinical symptoms are same like a pale shame and loose cell and hepatopancreatic damage, uh, and the pathology also we observe. The accurate result we may diagnostic from the proper laboratory. And bacillus also is ne- necrotic hepatopancreatitis, also the one of the disease causing agent. So it also same like loose shell and black piliopores. And we have another bacteria, it's called Streptococcus. Streptococcus also is reported in 2008 to 9. It is gram positive, and it caused the 100% mortality. And the hemolymph also, it looked like a whitish color. Uh, some people may think about it is caused by Bacillus series. And Lactococcus graviae is it mostly caused from the freshwater, and it is gram-positive white muscle-causing bacteria. It damages the discoloration, low level of continuous mortality. And Bacillus series. Bacillus series is commonly known as it is probiotic, but some mutant strain it caused the white patch disease. It reported in several publications and although the certain strains like can produce some strains may produce the toxin. Symptoms is like uh, appendage decoloration, internal mortality, the internal mortality. For Staphylococcus, the symptoms is like a standing syndrome. The shrimps swimming like in a standing position, so we can say like that. So the Lactococcus graviae is look like a whitish color, and Bacillus subtilis. Bacillus subtilis also it caused the white spot uh, uh, like a structure in the uh, shrimp, shrimp. So many of them they think about it is caused by the WSSV, but we have report about we report on Bacillus subtilis may cause the white spot, bacterial white spot. So it reported in 2000 in Malaysia, and another white ficus causing bacteria is Propogonium. It is anaerobic bacteria. So the symptom is look like a white fecal disease, a white fecal uh, white gut, but it caused from another bacteria. And rickettsia is astrotropy and pale color of EHP, yeah, hepatopancreasis. The whole animal look like whitish color. And systemic rickettsia, like bacteria, is milky disease. It causes milky disease. It identified from RGCA, Rajiv Gandhi Coastal Aquaculture Authority. Another disease is Mycobacterium, is a zoonotic human pathogen. It causes black spot uh, disease in shrimp. And another bacterium is called Aeromonas. Aeromonas is mostly occurs in the low salinity freshwaters culture. And next one is Pseudomonas. Pseudomonas is cause also the one of the disease causing agent. And Salmonella is ventral nerve cord disease. It infect the digestive tract, and lymphoid organs also it will damage, and its significant mortality will be occurs. And the next one is Halomonas is causing loose cell disease. It look like black spectrospora. And enterophagia is a black spot in body stalks. And filamentous bacterium, we have different type of filamentous bacterium. So some of the most of the bacteriums are like surface by fouling bacteria. So it commonly occurs in the hatchery. So before I like to share some sampling tips for a researcher or farmers. So the whenever we looking in looking the culture period. So whenever we collect a sample and we need to check the basic information from the pond. So whenever we collect the shrimp, we need to check the color of the animal, size variation, how much size variation is occurs, and deformity. Any uh, animal is bending or shortening or like that, we need to check. For color formation, we have different type of colors like normal animal and red in color, blue, yellow bands, dark yellow, some animal dark bioflock zinc also is available. So for red color means disease, it is maybe the disease from genetic stress. For blue shrimp and pale bright yellow shrimp and venomous tiger shrimp, due to the underfeeding and a mineral deficiency. And size variation also, I already explained about the how the animal size variation is happening. And this is the deformities. And we need to check shells, how the shell morphology is appearing. Like firstly, black spot. It is black spot means it is irregular or bilateral symmetric. And if irregular means disease, or mechanical means if we stock more animal into the small tank, it will it may be happen by physically. So this also we need to check. And another thing is external fouling, like Zuntanium, like maybe the worm infection or black matters. The white spot syndrome virus, about the white spots may appear in the animal. The molting stage, you need to check a few of things like intermolting or pre-molting. Pre-molting or post-molting or partial molting. This is what we need to check. And anterior also we need to check with the color cut, how the color means normal color, red color. Cut the antennae cut. We need to check with one cut or both antenna is cut. Look close to the body or how percentage of antennae is cutting. Tip black color or how the color changes is happening. And gills also we need to check every day. So normal gills and swelling are exposed to any infected gills or not. So that's also we need to check. And the color of the gills also we need to check, like black, brown, red, green, yellow. And the hepatopancreasis full slices occurs, or it may be the cut or any infection is there. So, and color of the hepatopancreas also we need to confirm. We need to check like chocolate color, white color, red color, black or yellow. So walking legs. So walking legs also are most important key points. So white leg and red leg and red near joints, black tips, yellow or necrotic. This is for a tiger chain. And abdomen also we need to check normal or muscles are good in nature, and red or necrotic, and splinter shape and deformities, head and abdomen gaps also we need to check. So for muscle, clear and transparent, and it may be sometimes white, and the body cramp also associated. Peliopods. Peliopods also we need to check, like it may it may be look like a normal or red, yellow, black roughness. So for black means it may be the NHP or loose shell or clear in water. This is black paleo code. For conclusion, note down all the negative points in the one page, and we need to check everyday health status. And it depends on the health status. We need to perform the dosage, and dosage of the treatment also we need to check. And so if we any doubts or any suggestion you need, so you need to discuss with your technical consultant. They will suggest you and recommend you how to perform the pond culture. Thank you. Soumya?
Yeah. Thank you, Ravi sir, for this detailed explanation on aquaculture disease in shrimp farming. Participants, you can check the chat box for the consulting services of Center for Ocean Research. Also, for more programs that are being conducted by Center for Ocean Research regarding the trainings, the workshops, and the hands-on programs, you can always check out the website, official website of Center for Ocean Research. And you can also check out the social media pages on LinkedIn and Instagram and Facebook for Center for Ocean Research. All these recordings, the lecture recordings, are available on YouTube, our live streaming channel. And sir, we have a few questions for you in the chat box.
Yes, madam.
Yeah. So there is a question, what is the meaning of qPCR? And there is a question saying that green colony is good or yellow colony is good for shrimp culture.
Okay, ma'am. Actually, the qPCR means it is kind of real-time PCR. Okay. So normal PCR, we can say only the positive or negative of pathogen. From our qPCR, we can see the how much infection is occurs in the body of the animal. So we need a check by a real-time PCR. This is very accurate study, more accurate study. So this is advanced method for detection of disease. Another one, the next question is green colony is good or yellow colony is good. The both the colonies are bad only. So the only thing is the concentration of the colony count of the what colony count is we need to take concern, because wherever we go, so all the ponds it have the yellow and green colony, but the concentration of the infection only we need to uh, check. So if some pond may go the more than 200 colony uh, per ml means we need to take more severe action on that pond. So if the yellow and green colony may go below 50 or something like that, so we no need to take much action of that form. So it is very common bacterial, you know. So it won't affect your culture. So it is very natural bacteria. So it will present in all over the world. So we cannot eradicate in our culture. So we have to maintain the concentration of the uh, uh, good, yellow and green colonies.
Thank you, sir.
Thank you.
If there are any more questions, please let us know through the chat box. Meanwhile, another announcement, admissions are open for MSc Marine Biology for the academic year 2026 to 2028 intake. The MSc program is a 2-year course, and the eligibility is any bachelor's degree in life sciences, zoology, botany, biotechnology, biochemistry, microbiology, environmental science, ecology, marine biology, or any life science oriented field with a minimum academic equivalent of 55% marks. The course fee will be 70,000 per year, and we have only limited seats available. The last date for application is 14th August. So we have a very few days left. The admission contact numbers are given in the chat box. For further information, you can reach out to our professors, Dr. Imbakandan, Dr. Tirunjana Sambandam, or Dr. Srinivasan, whose contacts are given in the chat box. Interested candidates are invited to apply and build your career in marine biology and ocean research and enter into the fascinating world of marine biotechnology. Official brochures and program details are available in the COR website and our social media channels. I request all the participants to kindly populate this word to your friends, juniors, and people in need. So with this, we come to an end of this session. The next session will be by Dr. D. Imbakandan, and we will join again at 2 p.m. using the same link. Thank you.